Hi, amazing science students, and welcome back to the Sound of Science. Today, we're going to be talking about cellular transport, the ins and outs of the cell. This is how things move in, things move out, and why certain things have to move in certain ways. So, it's important when we start talking about cell transport to understand the cell membrane and how the cell membrane works. So, the structure of the cell membrane is really made up of a couple of the different macromolecules we talked about. They're made up of lipids and transport proteins. So, lipids and proteins are the two macromolecules that we're really focused on when we talk about the membrane. Lipid part. So, remember the lipids are made up of fatty acids, right? If you look at this picture that I've given you, this is called the fluid mosaic model. You really should be able to recognize it. And you should be able to label it. That's a hint. Make sure you can label this. Um, when you look at it, you can actually see the little fatty acid tails of the lipids. And then, of course, the phosphate layer, the phosphate component of the phospholipid is there as well. The lipid portion is going to be hydrophobic, which means water fearing or water hating. The phosphate portion is going to be hydrophilic or water loving, or it's okay with water. So when you look at that, you can see the proteins sort of going through. Those are going to come into play when we start talking about the different types of transport into and out of the cell. But just for right now, make sure in your notes that you draw out this fluid mosaic model. It doesn't have to be exact, but just make sure that you understand that you have phospholipids, that there's two layers of them, and that the hydrophobic part's on the inside, hydrophilic's on the outside, and that you've got proteins that go through it sort of like straws or slides. The structure of the cell membrane makes it semi-permeable. That means that the lipid bilayer is actually able to move. It may be able to separate a little bit and to let small things sort of come through the membrane. Um, Things that are too big can't do that, obviously. So transport proteins are actually useful to let slightly larger things through, or things with ions and all that sort of stuff. So as we move through, just sort of pay attention to the types of things that are moving in different formats. Okay, now let's back it up for a second. We're going to talk about concentration. Not that you're focused. I want you to be focused. This, you don't necessarily need to write down all of this, but I do want you to write down the part where concentration is based on the amount of solute and the amount of solvent. So if you look at these two structures right here, you can see that one is much larger than the other, even though they have the same amount of solute, which is stuff. They don't have the same amount of solvent, so their concentration would be different. That's important to understand. For the cell, concentration is everything. Here's my Kool-Aid example. Again, you don't need to write this part down, just sort of listen for a minute. If I want to make Kool-Aid, I need it to be Kool-Aid concentration, right? If it's too watered down or too concentrated, too thick, it's not going to be Kool-Aid, okay? So if we take a single serve pack and we put it in a glass of water, it makes Kool-Aid. If we take the 2-quart pack, we put it in a pitcher of water, it makes Kool-Aid, right? Makes sense. But if we switch them, if we mix them up, then we end up with something that's not quite the same. We end up with really watered down Kool-Aid in the pitcher and really concentrated Kool-Aid in the glass. If we really, really mess up and we end up with something like this, the concentration is just wrong. And so we're going to come back to this example in a minute when I talk about osmosis. You'll see why. But just sort of keep this in mind that the concentration matters. The amount of solute per the amount of solvent is a big deal. Okay. Cells want to be at equilibrium. That's what makes cells happy. They really want to have the same concentration on both sides of the membrane. And there's a lot of different methods that cells will use to accomplish that. Make sure as you're taking the notes here that you understand what equilibrium is. So definitely write that down. Write down a definition of equilibrium. Okay, passive transport. There's three types of passive transport that we're going to talk about. And the first is diffusion. And let me back up. Passive transport just means that there's no energy required. This happens because it happens. It doesn't require any type of ATP or stored energy to make this happen. So, diffusion means that substances are going to move across the cell membrane, and it's based on what we refer to as a concentration gradient. So, the concentration gradient literally just means that you have a high concentration somewhere and a low concentration somewhere else. And think of it like a ramp. We're going to move down the ramp from a high concentration to a low concentration because that's what's going to get us to an equilibrium, an equal concentration. So if we look at this example here, this model, you'll see that we've got a high concentration outside of the cell, low concentration inside of the cell. So the materials are going to move through the cell membrane until they're equal, until we've reached equilibrium. Okay, some molecules, they're going to end up having to take a shortcut. So, with facilitated diffusion, we still don't require any energy. We are still moving along a concentration gradient to go from a high concentration to a low concentration, but the molecules can't just move through the cell membrane. Maybe they're ions, things that are charged. An ion's got a charge to it. They don't typically do well. They can't really move the through the cell membrane, through the lipid bilayer. You might also find things that are too large, may not be able to move through. So in this case, the cell wants them to move through, but they may not be able to do so on their own. And so we're going to end up using one of those transport proteins that we saw on the fluid mosaic model that I showed you in the very beginning. Transport proteins, think of them like a straw, or even because we're talking about a gradient, and I've told you the gradient's like a ramp, think of it like a slide. The molecules are actually going to go down the slide, and that's going to take them from outside to inside, or vice versa, from inside to outside. But regardless, facilitated diffusion goes with the concentration gradient, does not need energy, even though it's using proteins. So far I've talked about two types of passive transport, diffusion and facilitated diffusion. So let's move on to our last type of passive transport. And this is a special type of transport called osmosis. Osmosis is the movement of water across a membrane. So water is in a special category when it moves across the membrane. It actually can do so sometimes just across the membrane. Sometimes it actually uses those transport proteins called aquaporins or aquaporins. It'll actually move across then. So here's the deal with osmosis. So water is going to move across this membrane. And while it's also going from a high concentration to a low concentration, it's the concentration of water. So it's more of the, really, it's the amount of solvent per the amount of solvent. I've always found osmosis to be a little bit confusing when I think of it that way. So this is the way I want you to think of it. Okay. We're going to go back in our slides to our Kool-Aid example. Now, if we're looking at our Kool-Aid example where we've really messed it up and we have our large packet in a little tiny cup, how can we get a better concentration? How do we fix that concentration? We're going to add more water, right? More water will actually make the concentration better. It will help dilute that too strong Kool-Aid. Cells are going to do the same thing. When you have something like a sucrose molecule that's huge and it's difficult for it to move, water is actually going to try to dilute the side that has the sucrose. It's going to try to dilute the side with the amount of salt, the NaCl. It's going to try to dilute wherever it can to help encourage equilibrium. Okay, so when we think of osmosis, we think of how water is moving. If you can figure out the direction that the diffusion would go, which direction the stuff wants to move, then osmosis is going to occur in the opposite direction. If stuff is moving into the cell, water will be moving out of the cell. And that's what I want you to keep in mind. Okay. So for osmosis, you need to know what it is. You need to understand how water moves, that it's going the opposite of diffusion. And then I need you to take a look at this little item that's here on our left. So, when we put cells, particularly human cells, or really any cell, into different environments, the water is going to move in or move out um, really based on the equilibrium. So, in that first example, all the way to the left, what we've done is we've placed the cell into a hypertonic situation or a hypertonic environment. When we do that, like for instance, seawater or salt water, the water inside the cell is going to try to dilute the environment so that the cell will feel like it's at equilibrium. It can't, but the cell will literally shrivel itself up, getting rid of all of its water to try to reach that equilibrium. So, in a hypertonic solution like salt water, the cell is going to do what we call crenate, with an N, not an M, crenate or shrivel up. In a hypotonic solution, I want you to think of that word hypo. It makes an O sound, right? In a hypotonic solution, such as pure water, where the high concentration of stuff is inside the cell, water will actually move into the cell and the cell will swell up like an O. So that's where we get hypotonic. And then our final one is isotonic. An isotonic solution is when it's really at equilibrium. It's the same concentration of stuff inside and outside. And that means that water's going to freely move back and forth. And that's just what happens all the time anyway. Stuff moves back and forth when it's at an equilibrium, it's not at a standstill. But we will have zero net movement, which means it's not really going in one direction or the other, it's moving in both. So, situations that we would think of hypotonic solutions. In a human, that would be when somebody has something like water toxicity. They've taken in so much water that there's not enough solute outside of their blood cells, and their blood cells will actually swell up until they burst. They'll pop open. This is why when you get an IV at the hospital, let's say that you're sick and you're dehydrated, they give you an IV. They always give you something that's a saline solution, has a little bit of salt in it. And it's just enough so that your cells don't swell up like that. They don't want them to do that. Hypertonic solution would be, for instance, if you were drinking seawater. This is why we don't do that. If you drink seawater, then your cells are actually going to continue to dehydrate because you're taking in so much sodium, and the concentration will just get worse and worse and worse. And so you actually are doing more harm than good. Isotonic is what our body maintains to be at. That's what it really wants to be at all the time. So hopefully this has made sense. I know that osmosis can be a little bit confusing, but just remember it's the opposite of diffusion when it comes to movement and determining where things are going to move. Okay, so now it's time for us to talk about active transport. Active transport means that it needs energy. It has to have energy. And typically that energy is going to be in the form of ATP. Remember, we talked about ATP when we talked about cellular respiration and photosynthesis, right? Cellular respiration makes ATP, stored energy. So, active transport, like passive transport, there are three types that we're going to talk about. And as we move through and we look at these three types, I want you to kind of keep in mind the different situations where this might occur. Okay, so the first type we're going to talk about, we're actually going to talk about two at a time, is exo and endocytosis. Exocytosis is when the cell has to release something, and so it's going to allow something to exit, hence exocytosis. This is something that we're going to see with like the Golgi apparatus. Let's say that some proteins have been made by the cell, and those proteins, those enzymes, whatever they happen to be, need to be transported outside of the cell membrane. Well, proteins are pretty daggum big, and they're not just going to slide through the cell membrane. So the Golgi apparatus is going to form a little vesicle. It's going to carry the proteins to the edge, and then it's going to just and pop it open and allow those proteins to be released. They're going to be secreted. Endocytosis is when something comes up and the cell needs to consume it. And so it'll do so by surrounding it and creating a little vesicle that it can actually use to digest it. Think of amoebas. Which hopefully you guys will kind of remember vaguely from many years ago. Remember, amoebas are those protists. They're able to sort of move by like Bob the Blob from Monsters versus Aliens. So they'll actually surround their food with their little amoeba parts and create that little pocket. They'll digest it, and that's how they're going to get their nutrients. So that's an amoeba. The third type of active transport we're going to talk about is use of a protein pump. Protein pumps are exactly what they sound like. It is actually going to pump items against the concentration gradient. And that is really the thing that we see with all active transport. It needs energy because we're going against the gradient. Just like if you are on a bike or a skateboard and you're at the top of a hill, it doesn't require a whole lot of energy for you to move down that hill. But if you're on a bike or a skateboard and you've got to go up a hill from the bottom, you're going to need to put some energy in. And that's what cells are doing. They're putting in the energy to go against the gradient. So in a protein pump, we're using ATP to actually change the shape of the protein so that it can go against the gradient. Okay, so a side note, which is an important side note. So surface area to volume ratio. The more surface area you have for the total volume, the easier it is to diffuse across the membrane. So this means that the larger a cell gets, the less effective diffusion and osmosis will be. So cells need to stay small so that they can actually diffuse items all the way inside. And if you look at this picture I've given you, the places where the gelatinous cubes are pink, that's where things can't diffuse to. That's where it can't reach. And so the tiny little cells, they have much better rates of diffusion and much better diffusion ability than larger cells. That's why cells can't be big. They can't be big cells or they won't work. They don't get the solutes they need. They don't get the nutrients they need. They don't get the water they need. Other things that to keep in mind, like your organelles, like your mitochondria, it uses membranes. It needs surface area. And so it actually is going to have lots and lots of little folded up membranes. That's the things that look like the squiggle. Those folded up membranes increase the surface area. If you think about your own body, remember, science is patterns that we see over and over again. If you think about your own body, think about your intestines and how many folds they have. And that's because we're trying to absorb all of the nutrients we can out of our food before it passes out as waste. Okay, cell specialization. Unicellular organisms have to maintain homeostasis, right? And they're going to do this using all sorts of types of transport, right? So they maintain homeostasis. They're really not super specialized. Multicellular organisms, on the other hand, also use transport, but they're much more specialized in their cells. Because we have so many different types of cells, our cells actually have to send signals to each other. They have to coordinate. That way everything can sort of happen like it's supposed to. And our cells do this by using signals and receptors. So we'll actually have receptors just sort of hanging out on our cell membranes, and we will oftentimes have hormones, or enzymes or proteins. Um, these sorts of things will actually come and they will interact with those receptors and let our cells know what's supposed to be happening or what they need to do. And that's one of the things that we see, like, for instance, when you get hungry, you actually will start, if you don't eat, you'll start releasing a chemical called glycogen. And that glycogen will start being broken down into glucose so that your body can use it for cellular respiration. And we do that because our body actually sends signals in response to our blood sugar dropping that lets our body know, like, hey, we need a little sugar here. And so your body is immediately going to start using that glycogen. It's going to start breaking it down for glucose. And it's going to give you some signals that your blood sugar is getting a little low and you need to eat something. You need to put some food in your system. And you'll notice that your body actually gets trained that as soon as you start eating, you'll already start feeling better, even though the glucose hasn't hit yet. As you are growing and developing, the cells are actually going to specialize really, really quickly. It's not very long as an embryo that we have sort of all the same cells, all little stem cells. We actually start to specialize really fast. Every cell has the same DNA. And so, because they have the same DNA, every cell is sort of capable of being any cell it needs to be. But again, we're going to use these signals to determine which types of cells develop where. Okay, so here's just an example of some of these cells, the specialization that we might see, the different types of cells. And this is one of those reasons why I always ask the question, are all cells round? Are all animal cells round? And the answer is no. Cells actually have many different shapes. So just sort of something to keep in mind. Anyway, hopefully that helped you understand cellular transport. Hopefully you got all the information written down that you needed. And I look forward to seeing you next time.